137.276. If you're off on your last digit, I don't care. That's just something your calculator did. All right? Know how to calculate osmolarity. You may see this again, but not in this format. All right? Number 29. Simple squamous epithelium is found where? Bowman's capsule. Where else?
Alveoli.
Alveoli. Where else?
Serous membrane.
Where?
Serous membrane.
Don't need to be that technical. How about the lungs?
That's alveoli, right?
Alveoli, yeah. You can just put lungs. Blood vessels,
Yes.
like capillaries. Okay? If you put it in the box, you need to give me the function. So for Bowman's capsule, what is the function of this?
Filtration.
Filtration. It's also diffusion. Both of those would need to be there. If you gave me the lungs, it would just be diffusion.
You'd say gas exchange.
Diffusion. Use your big words.
What if we put
You're getting
both?
paid for your big words. Okay? Well, that's what you're getting. That's why you start learning them now.
If,
Yeah.
if we can put more than one.
Yeah, you can put more than one. But remember, if you put more than one for location, give me more than one function.
Okay.
What about stratified
The
squamous?
skin.
Skin and esophagus.
Vagina.
Vagina.
There we go.
Hey,
Hey.
Humans always have this word.
And its function?
nothing wrong with that.
Protection.
Nothing wrong with that.
33 to 36. 33, what's the cell type?
Alpha
And
cells.
what does it make?
Glucagon.
And number 35? And what does it make?
Insulin.
Okay. From this, you need to know the hormones that are produced by each endocrine organ, each layer of the endocrine organ, each cell type of the endocrine organ, because that is what was given to you in your lab. Right? A feedback loop, does it go in both directions?
Yes.
Yes. How do you know that? Because it gives you a range. If you got a plus and minus, it means you're going both ways. Everybody know where to find the information for this feedback loop.
Yeah.
One side's using insulin, the other side's using glucagon. All right. When you go back, your set point is always in the middle. But when you go back to
The range.
the range, something in the range. All right? Describe the posterior pituitary gland in detail. Does the hypothalamus control this gland? If so, how? What are the target organs for the posterior pituitary? This is a long answer, and yes, you do need to be specific. If you want to do it in a flow chart, you can do that also, if you can think of a way to do it. So what does the posterior pituitary control?
Controls the
Body water regulation.
Water regulation, and what hormone is that?
ADH or antidiuretic.
Vasopressin?
Yeah.
Okay, that one. Yeah. And what's the other one?
Oxytocin.
Oxytocin. What's the target organ for the oxytocin?
Ovaries.
ovaries.
And mammary glands.
And
You can also put kidney, because that's a target organ for the vasopressin. If you want to get real technical and put down exactly where in the kidney, you can, but just kidney is fine. Ducts or collection tubules.
when it says describe it in detail, is that what we just did, or what else is it looking for with that kind of question?
With that kind of a question, you need to mention something about the hypothalamus.
That it's connected to it, or
How is it connected to it?
Neurons.
directly?
Neuro,
By
neurosecretion.
Neurons?
neurons. Axons of the neurons in the hypothalamus go directly to the posterior pituitary. The hormone is secreted down the enzyme, so that needs to be in there. It can be as short as you want, but all those points need to be in there. Which is opposed to the anterior pituitary. So how is the anterior pituitary connected to the hypothalamus?
Blood vessels.
What's the name of the blood vessels? A portal system, use your words. A portal system that begins and ends in capillaries. What is the hypothalamus releasing in the portal system?
Release.
Releasing factors.
No. It calls them release hormones in the, wait.
Whatever.
Okay,
As
fast.
long as that release is in there,
Right.
you're good. And there's a whole bunch of hormones for that. You need to know all those hormones, all right? Pretty much, that's why it's the master gland, it controls everything.
Yeah.
The graph. For the graph, you have two y-axes. You can put temperature on one side and hematocrit on the other. Don't forget your units. And then at the bottom is time. Time is measured in
Days.
days or weekdays or date, depending on what you put there. All right? I'll go over graphing in a minute. Interpret your graph. What's happening to the patient based on these two measures of body function?
Temperature's going down,
What's happening to the hematocrit?
it's going down and coming back up.
Yeah, at a certain point, it comes back up. So once the temperature reaches a certain point, it will kick in and hematocrit will start to go back up again. So put into words what you're seeing happening and the relationship that you see between temperature and hematocrit. All right? The lectin chart you will have access to in the handouts that you should have on the bench. If it's not there, you ask me for it, okay? So what's in here is a periodic table. You have the lectin chart. You have a normal karyotype, which means I can give you an abnormal karyotype. And you also have the genetic code. All right? Will you be using all of them? Maybe, maybe not. Probably not all of them. All right. Which glycoprotein did limulus polyphemus lectin bind on the surface of the red blood cells? That's where you would use, and actually you have a clue. It's either galactose or sialic acid, but you go to your chart and you see which sugar it is specific for. So it is sialic acid, all right? What was the control in this experiment? So this is where you need to know your conditions. So what are the two controls?
41 and 43.
41 and 43, but you need to tell me what is on the slide. Okay, so write it out. 41 is a negative control. It has no cells in it. It only has has cells. It doesn't have lectin in it. If it doesn't have lectin, you cannot have a reaction. Whereas 43 has everything. That is the perfect condition for a reaction to occur. So that is your positive control. Given the following information, calculate the concentration of an unknown protein using Beer's law. So she will give you an absorbance. She will give you the concentration of the standard and the absorbance of the standard. So absorbance of the unknown, concentration of the standard, and absorbance of the standard. And those, you should use Beer's law to calculate what the concentration is of the unknown. So what is that concentration? Anybody do that part?
Get that far.
What?
Didn't get that far.
54 grams per deciliter. Make sure to put your units in. Just don't give me a number, give me the units also. Make a prediction based on your knowledge of what Beer's law says. What is the expected concentration of a protein standard that has an absorbance of 0.04? You do the same thing that you did here. All right? It's a standard, yes, but you have another standard to compare to. The answer is 6 grams per deciliter. Under what conditions might hypoglycemia occur? Tell a story that is a reasonable possibility. If you want to put that story into bullet points, that is fine. Okay? but I must see a few things, like if you're hypoglycemic, what did the patient do?
You got lost in the desert.
Well, what happens when you get lost in the desert?
You don't have
That's
any.
You don't eat. That's all you need. Patient did not eat. All right? Then what else would you put in there?
Some,
Right, if the patient is diabetic and didn't take his insulin, that's also gonna happen. So there's two conditions you can put in there.
and then could you put symptoms in there as well?
No, it's only asking you for conditions.
Okay.
What, condition is not a symptom. Here, condition is what did he do?
Do to cause it.
Mhm. What are the usual remedies?
Food.
Sugar.
Sugar intake.
Glucose, okay?
Orange juice.
Orange juice.
Yay.
Explain how our goldfish experiment helps you to understand this clinical condition. What was supposed to happen to the goldfish when he was in
The insulin.
hypoglycemic? Yeah, when he was in the
He's supposed to slow down,
Slowly die.
lethargic.
slowly die. He's supposed to become lethargic, everything a human patient would do. Activity decreases, respiration decreases, okay, he just kind of sank to the bottom. Yeah. Insulin is a peptide hormone. Describe its solubility in an aqueous solution and why. Is it soluble or insoluble?
It's soluble.
Soluble. Why?
Polar.
It's
It's not lipid based.
Amino acids. What do acid imply?
Hydrogen.
It has a charge. It is polar. Anytime you see acid or base, you know there's a charge there, which means it is soluble in water. Like dissolves like. And why? Because water is polar. Again, you can put in bullet points, but I need to see those three things. A student in the lab said that the hormone diffused into cells. What would you say to her?
no,
No. Why?
because hormones don't diffuse into cells. They latch on to
Use your words.
Go into the bloodstream.
Receptors. So hormones bind to receptors. If the hormone is nonpolar, in which case it is lipophilic, then it will go straight through. Why?
because they're both.
Lipophilic.
Because the plasma membrane is mostly lipids. So steroid hormones, that's
Yes.
why they're so potent. They are lipophilic. They go straight through the membrane, don't need a receptor, go straight through into the nucleus and cause change. All right? So be careful with anything that's lipid-based.